Greetings and welcome to our poster. This is Yatindrapravanan Narasimhan from final year BTech Bioinformatics, Shastra deemed to be University, Tanjavur, India. I and my team have worked on a project that extracts certain molecular features from the molecular dynamics simulation trajectory data. Further, our goal is to train a classifier based on this data that predicts if an input ligand is an agonist or an antagonist for a particular receptor. We have used the ligand binding domain of the human androgen receptor and its known interacting ligands annotated with agonist or antagonist labels in PubChem. To get macromolecule structures, we got a true positive and a true negative structure of the ligand binding domain of AR from the RCSB PDB database. 4HLW is AR bound to testosterone, and this is taken as the true positive to dock the agonist molecules onto, and 2OZ7 is AR bound to cyproterone acetate, which is the only FDA approved drug which has antagonist activity in AR. So the antagonists were docked to the true negative structures. We ended up with 455 agonist labeled ligands in total and 981 antagonists from the PubChem. So to avoid bias towards antagonists in our datasets, we filtered the 981 antagonists based on IC50 values and got the 455 antagonists of the least magnitude. To ensure that there was no structural bias within the 455 antagonists, we performed k-means clustering based on the structural similarity with the true negative structure using the Tanimoto coefficient, and observed that we have a healthy representation from all structures. Further, we performed an exhaustive blind docking using the Lamashkian genetic algorithm of AutoDock 4.2.6 on all 910 ligands with the two macromolecules. The distribution of the least binding energies of the antagonist AR complexes is plotted as a histogram here in figure 5. There were certain missing atoms of residues in the N and C terminal regions of the receptor structures which needed to be modeled before simulating the docked complexes. So, the FASTA sequences of these receptors were modeled in Rosetta CM of the Roberta server. The Ramchandran plot was generated for the model structures and no outliers were observed, with 100% of the residues falling in the allowed region. The model structures were aligned to PDB structures, and the rotamers of the active site residues were observed to be in the same orientation. The alignment of the whole molecule and also the rotamers of the active site residues are shown here in figures 6 and 7. The Gromax Amber force field Amber 99 SB 2010 was used to simulate the complexes. The Python wrapper AC pipe was used to run anti-chamber to generate the Amber specific files for the ligands. And then the equilibration step was run for 100 picoseconds, followed by a 20 nanosecond production step. We concluded that 20 nanoseconds were enough to study the dynamics of the complexes using the RMSD and potential plots. There was no significant deviation from the start or no significant deviation after a point of time in these plots. So the same plots are shown here in figure 8 for the true positive receptor bound to the testosterone. Figure A of 9 shows the 20 features that we extracted from the simulation trajectory data. The criteria were just that the data was extractable, as in terms of not having higher dimensionalities. We ended up with the time series data that has all these 20 features listed for each time step for a complex. So for each complex, which are essentially separate CSV files, were decomposed into a single CSV file having one complex per row. We used the TSFresh tool for this purpose that calculates approximately 749 features for a single feature, such as the mean, standard deviation, min-max value, and so on for say a feature HBnum, which says about the number of hydrogen bonds we have. So we took a proof of concept dataset having 30 agonist and antagonist AR complexes. So we ended up with 15,677 features in total. Figure 10 here shows an illustration of the simulated complexes, the top row being the agonist complexes and the bottom row the antagonist complexes. The leftmost figures of both the rows show the control structures, that is AR bound to testosterone and cyproterone acetate respectively. So far we have simulated 360 complexes for 20 nanoseconds, that is 7.2 microseconds data. Figure 11 here shows the selected features from TSFresh output by the ensemble feature selection tool. A list of 13 features with an EFS score greater than 0.5 was considered significant. Further, we plan to extract and select features for all 455 ligands in both sets and train a classifier. The scope of such a work is restricted in our case, but not limited to the androgen receptor and its ligands. The primary aim here is to build a method, as such features from dynamics data either do not exist in entirety or with sanctity in current literature. Thanks for listening. Shubha Dinamastu. Have a good day.